No question?
So you can start.
So no questions. Kedar, were you able to fix your problem? Can you ask your question now? Yes, you have raised your hand. You may ask your question, Kedar. Can you please unmute and ask your question?
Hello.
Yeah, Kedar, I can hear you. Go ahead.
Yeah, sorry, Suryadeep. Actually, we were disabled to like unmute, and also the chat was disabled. So I
Oh,
think
is
it was
that
not
so?
possible to. Yeah.
Kedar,
Okay,
can't
okay.
you unmute yourself?
Can you try again? You are muted again.
You should, because that's why you spoke now, right?
No, I unmuted him. If I do this, I think now he can speak. Try speaking,
Okay.
Kedar.
Hello.
Yeah, you can speak now.
Hello.
So some
Yeah. So
problem maybe.
actually,
Yeah, go ahead, Kedar.
yeah. So actually, I was not allowed to unmute earlier, and then
Kedar,
for
sorry, there was some issue with Zoom at the moment. That's why recording also stopped. Can you please try it in chat now? That should work, hopefully.
Can you try writing to the chat, Kedar? If it's not working, please raise your hand.
That should work. There was some error with the platform itself. Anyone kindly post something. I guess. So did you receive text, right?
Yes, I will. Okay. And you can also unmute yourself now, right, Kedar? Can you try doing that?
Mm-hmm.
Maybe the unmuting is still not.
Unmuting is still a issue.
Unmute is disabled. Okay, no problem. At least for today's class, then, if any of you have a question, just raise your hand. I can unmute you from here.
Now, so did it. Let us confirm. Kedar, can you please try now?
yeah, now it works.
There was some issue in the platform. Okay, sorry, guys.
Okay, no
Let's
problem.
continue.
Yes. So, Kedar, you had questions, right?
Yeah, I was just asking about the p-adjusted value. I think
Correct.
it's in the previous slide.
Correct. So, do you remember we talked about FDR correction yesterday? False discovery rate correction,
Yeah.
right? So that is what the p-adjusted value is. So you remember we calculated the p-value using Waltz test, right?
Yes.
Yes. So that is the p-value that is there in this table. Let me show you once. You'll probably be able to understand much better then. My screen is visible, right, Kedar? Yes. Is my screen visible?
Yes, your screen is visible.
Yeah.
Kedar, still the problem is existing?
No, I can unmute.
Oh. So just take a look at this. This p-value is the one that is coming from the Waltz test that you're performing only for this gene, RPL26, and so on and so forth, all of this. Okay. Now, if you remember, we talked about false positives, right? You can end up with 1,000 false positives, right? If you take a significance level of 0.05, right? So that is what, 5% times 20,000. So you can get around what, 1,000 false positives. So in order to fix for that, what we do is we do something known as FDR correction or false discovery rate correction, where we employ a method, at least DC2 employs a method known as Benjamin-Hochberg correction method. What it does is it does something called multiple testing. So not only does it calculate a p-value separately for this gene, it also calculates a p-value taking into account the fact that we are doing this Waltz test for 13,000 other genes as well. And that p-value is the p-adjusted value. Okay, this is the Benjamin-Hochberg adjusted p-value. And ideally, statistically, this is the one that you should use for your reference. All your cutoffs should be based on p-adjusted value. But as I said, sometimes you just have to ignore the p-adjusted value and go for the p-value, because you will end up with very less number of differentially expressed genes if you use the p-adjusted value, especially when it comes to data which is of quality as bad as this. Okay, this is bad quality, right? And that is why it is okay for you to use p-value and not p-adjusted. Although ideally, in the most ideal case, in most cases, you should always use p-adjusted. This is the correct one. Okay, but for our purpose, for this analysis purpose, we are going to be using p-value. Okay, because otherwise we were getting only 40 differentially expressed genes, right? That is what we saw. Okay, does that answer your question, Ketan?
Yeah, so it means it's like a more stricter, or I should say, yeah.
Correct.
Okay,
It's a
got it.
stricter version of p-value, basically, p-adjusted. And it's a
Okay,
stricter version as well as it is the correct version. Okay. We are using p-value
yeah,
because strict is not working for our data, as our data is not very clean. Our data is not that high quality. With high quality data, p-adjusted value works best always. Okay. And I could have easily shown you a data that clusters very well and very high quality data, but then that would defeat the purpose. Okay, I want to show you guys that although that life is not all about sunshine and butterflies, okay, data will be bad, data will be dirty. It is you who has to adapt to it and make sense of it anyway. Okay, so these are some things that people end up doing. So you might as well learn these as well. Okay, that is the whole point. Okay, with that said, so this is the heat map of the top 25 upregulated genes, right? Very similarly, I can also plot the heat map for the top 25 downregulated genes. I have to first create the heat map data down as the matrix. I'll go ahead and run it. The other stuff is all the same, except for this parameter, which is clustering method. Here you can see that I have not used a clustering method. Okay, but here I have. I will tell you the reason why in a minute. I will first run this, and you can see again, I have very distinct patterns, wherein these are of the top 25 downregulated genes. So in LAA, they are all blue, so they are downregulated as compared to HC. But in HC, they are all, not all, but we have a significant amount of red. That means these guys are upregulated as compared to LAA. Okay. Now, I mean, the interpretation is the same as that of the upregulated ones. Okay, so I'm not going into the details of that, but what is very interesting here to notice is what happens if I kind of remove this. Okay, so if I kind of remove this particular parameter and I run this, this is something very interesting. Now you see the difference between this, right, and this. This HC2, which was an outlier, if you remember, has been detached from this, from its HC1 and HC3, right? But I can still clearly see the red here. So ideally it should work, and ideally it should cluster very well with HC1 and HC3, right? But this is not happening. And that is because of the clustering strategy. Okay. So the clustering strategy that by default happens in the p-heatmap function, it does the clustering based on Euclidean distances. Okay. That is one of the most basically the Cartesian coordinate, in the Cartesian coordinate plane, the basically the 3D the distance matrix. That is what the Euclidean distance is, right? So it's just a normal distance. In case of the downregulated genes, what I did was I used something known as the Ward's distance to make it work. Okay, so if I again go ahead and write here clustering method, and it will be Ward dot d. Right? If I go ahead and run this again, I probably wrote the name wrong here. Do something else. Yes. It was correct too. Something okay, something was anyway. We go ahead and run this. Here now again you can see that this in this case, right, here the clustering did not work well because it was Euclidean distance. But if I'm using Ward's distance version 2, here the clustering is working very well. Okay, now again, these are things that is not possible for you to know just like that, okay, what will work and what will not work on my data. Okay, so this is a lot of trial and error you have to do. So clustering method has several parameters you can give to it. You can give any kinds of distances to it. You can give Manhattan distances, you can give Ward's distances, you can give Euclidean distances, and every other form of distance metric that you can think of. Okay, what will work is something that you have to figure out by trial and error. But you can figure out kind of why Ward's distance works well. That is because Ward's distance again is not very sensitive to outliers. Okay, it is not sensitive to outliers. It does not change drastically, even though there are outliers in our data, which is true because if you think about it, HC2 is an outlier. Okay, so HC2 could be causing us a few troubles. Yes, LA2 could have also caused us a few troubles, but it did not. HC2 did. Again, that is completely on the math. We are not going into the math. If we were to work it out on paper and figure out the math, probably would have figured out what is the problem that is happening. But again, then again, we don't need to do that specifically. So it's absolutely fine. Okay. So we know HC2 was an outlier, and there was always a chance this was gonna make life difficult for us, which is exactly what it did here, right? But you can actually fix it by fixing the clustering method. Okay, using a method that is not as sensitive to outliers, something which is Ward's distance or Ward's declude distance. Okay, this is not as sensitive to outliers, so therefore, even though there is an outlier like HC2, if you use Ward D2 method, HC2 will cluster properly well with the other healthy countries. Okay, and the pattern here is again pretty clear. Okay, because you see red very frequent here, but here it is almost okay. I hope this is clear to all of you. Okay, as I said, for each and every one of these, just size it properly and then hit the export button here and save it as an image or a PDF, however you want. Okay, you can also directly copy it or click save image from here, whatever you do. Just make sure you have all the writings properly visible and so that you can get these pictures out. Okay. So is everybody clear on the heat maps? Do people understand what a heat map is, how you interpret it, what is the purpose of the heat map? Yes. Others? Any questions on the heat map? Okay. So now we will understand one more type of visualization, which is known as a volcano plot. Okay. So for a volcano plot, the first thing that we need to do is create a column in our results data frame as our negative log p-value. Okay. So p-value, again, it is a very small range, right? P-value ranges from anywhere between 10 to the power -7 to 0.04, 0.05, like that, right? So it is a very small range. Visualization in that very small range becomes very difficult. So you kind of perform a minus log 10 transformation on the p-value to kind of widen the range. Okay, if your p-value is within this small range like this, you kind of widen it by performing a minus log 10 transformation. Okay, and you create this extra column, which is the negative log p value in your results data. Once you have done that, you add another column known as expression so that you can color each data point properly. So how you do that is you use this if else function. So what it does is basically you give the clause here, you give the condition here. Okay, so the condition, so this is read as res tf $log2 f change if it is greater than or equal to 0.5 and the p-value is less than or equal to 0.05, you add upregulated for that particular row. Okay. Again, if else, res df $log2 full change if it is less than or equal to minus 0.5 and p-value is less than or equal to 0.05, it will write down regulated. It will annotate it as down regulated. Okay. And else, that is if it does not match any of these two conditions, when would that be? If the p-value is greater than 0.05 or if the log2 fold change is anywhere between 0.5 and minus 0.5. So for example, a log2 fold change of 0.2 or minus 0.2, 0.1 or minus 0.1, such log2 fold changes would be assigned as not significant. Okay. So if I go ahead and run this, you can see that in resdf, I now have this negative log p value column and the expression column, which has upregulated, downregulated, as well as somewhere along the line, not significant. Okay. So now I will plot something known as the volcano plot. Again, I'm going to be using ggplot here to plot the volcano plot. I will first create the volcano plot object. Then I will call ggplot, right? To it, the first thing I have to do is obviously give the data. The data is resdf. The x-axis is going to be your log2 fold change, and the y-axis is going to be your negative log p value. You are going to color each and every data frame based on expression. That is whether it is upregulated, downregulated, or not significant. Okay. The rest of them are kind of same geo point, you guys remember, right? So that it uses points for each data point. Excuse me. This is the color scale that I'm going to use, right? Red, black, and blue for upregulated, not significant, and downregulated. Okay. Now what I'm going to do is I'm going to put a vertical line, hence why 𝐿 line, and horizontal line, hence why 𝐿H line. Okay. So my vertical line will be at my log FC cutoffs. That is why the XY coordinate for the vertical line is minus 0.5 and 0.5. So my vertical line will go through minus 0.5, 0.5. Okay, the line type I want as dashed, and I want the line color to be dark green. That is all there is to it, right? My horizontal line will be along minus log 10, 0.05, because that is my p-value cutoff. 0.05 is my p-value cutoff, and if you remember, my y-axis is minus log 10 of this. That is the negative log p-value. So that is why minus log 10 of my 0.05. So this is basically, I am putting two dashed lines, right? I am putting two dashed lines as a to indicate a zone of significance, basically. Okay. Then this labs function basically adds labels. I'm adding the main title, right? This is volcano plot. And then I'm adding the X label and the Y label. So it will label the X and Y axis accordingly. I will go ahead and run this. This will create the volcano plot object. Now I will add the title based on whatever I want. So I have put differential gene expression analysis of control versus large artery atherosclerosis. If I go ahead and run that, it should come up here. Yes, you can see this volcano plot coming up here. The legend already comes here when you use ggplot. That is a very good thing about ggplot. All the down-regulated genes are marked in red. All the upregulated genes are marked in blue. And the ones which are not significant are marked in black. Right? You can see this gray line here. Right? Anything to the right of it are upregulated. Anything to the right of the gray line and up of the gray line are upregulated. Anything to the left of the gray line and up of the gray line are down-regulated. Anything below this vertical gray line, right? They are not significant, obviously, and anything which is, that is why this horizontal line, and anything to the left of this, anything to the right of this are upregulated, downregulated. Anything in the middle of these two horizontal lines, again, they are also not kind of this thing, not significant. Although we don't see we don't see a lot here. We only see quite a few here, which are also below this gray line, so they are kind of downregulated, not significant. So this is again another very famous visualization that helps you visualize your differential gene expression analysis. That out of all of these genes that you see here, so you see this as these as continuous blobs, right? You don't see them as separate points, but that is because the points are very overlapping on each other. So out of all the 13,000 genes, these are the ones which are upregulated. These are the ones which are downregulated. Your x-axis is your log2 fold change, so it's pretty easy to understand. Anything to the right of 0, that means positive fold change, so that would be upregulated. Anything to the left of 0, that means again positive fold change, so negative fold change, so downregulated. And this is kind of your p-value cutoff. Anything above this is statistically significant. So this is how you can kind of interpret a volcano plot. Okay, it kind of looks like a volcano erupting. That is why it's called a volcano plot. Okay, you can also use this complex piece of code using again ggplot. It allows you to actually label the genes, and that becomes very handy if you want to investigate it further. So I won't explain how this works again, because again, this is syntax. You kind of have to memorize it. There is no way to kind of actually just tell you guys and you will remember it. No, that won't happen. It's something you have to memorize. And honestly speaking, guys, even I don't remember how to write this. Okay, I have to always refer to the manuals, right? And that is absolutely fine. It is not possible for you to remember all of them. As long as you remember most of it and you have the ideas, it is absolutely fine. So I will simply select this entire part and run it, right? And okay, there was some error here. Okay, some form of error happened here. I have to probably fix this script. Something would have happened. Okay, anyway, I will fix this script. No worries. Basically, here what will happen is the gene names will also pop up. Okay, with arrows to the data points, that you can figure out what each gene is here. Okay, in the HR script, I think it kind of works. So I'll be able to show you there. Even if I can't, I will fix this maybe right now after the class. I don't want to take up time in the class because I have to first figure out why this error is happening. Okay. You know, say right, how it makes sense. Ah, 25 and then control. Okay, some error might have happened here. So I'll try to figure that out, what exactly happened. So, this one. Oops, this. Give the wrong variable name. Yes. Now you can see I will actually zoom it. Maybe we can see it even better. Yes. Now you can see most of the genes are now named. Okay, all of them are not named because of overlapping issues and all. So again, these are again some what can I say, limitations with respect to the system. We try to name all of them, but again, it does not become possible to name all of them. But at least most of them are named, so that is good. Okay. So yeah, this is something that is there. Okay. So something. Okay. So this is all there is. But now I think the labeling is a bit messed up actually because I see some repeats. I'll probably have to fix the labeling. I don't think the labeling is quite correct. But anyway, it is possible to label it. Okay. Using this. So, but again, in most cases in papers, you will see people don't bother labeling. They just go ahead with the default volcano plot, and whatever are the values, they kind of figure it out, and they kind of use the alerts, right? So they kind of figure out what each of them is and then label only the important ones manually. That is also something you can do. Again, these are all personal preferences. Whatever you want to do, you can. That's absolutely up. Okay. So this is about volcano plots. Does everybody understand what a volcano plot is? How to interpret it? Can anybody confirm? Okay, one of you. Others, can you please confirm if you understood what a volcano plot is? Okay. Others, any questions on the volcano plot? What it is? Okay. No questions. Okay. Very good. Okay. So that is all we have for DEC2. Okay. So with this, we come to the end of DEC2. For HR, as I said, the script is 80% the same, except for a very small portion where we actually perform the HR analysis. So that is something we will do in tomorrow's class. Okay, we will perform analysis using HR script. Okay, we have a small amount of time to go through the HR's normalization procedure. Okay, so what we do is we can quickly go through the normalization. Can be it's how much it is 22 slides. I don't think we will be able to finish 22 slides in 15 minutes. So what we'll do is it will take around half an hour to finish the HR theory. Okay. And it will take another half an hour or 15 minutes to finish HR's practical. Okay, because the script and the plots are the same, so I won't be explaining the plots again. I'll simply run the script and show to you guys how it's working. Okay, so we can do that in tomorrow's class. Okay, and the day after tomorrow, that is 24th, we'll have a discussion on the mini project, and I'll show you guys something known as enrichment analysis, which actually helps you perform some downstream analysis, which actually helps you make sense, make biological sense of your differentially expressed genes. So once you end up with a list of differentially expressed genes, you have to do something with it. Right? You cannot just sit on. So what you can do, one of the things that you can do, I will show you. This is enrichment analysis. That will be day after tomorrow. So this will be our agenda for the next 2 days. Okay. With that said, I think we can wrap up for the day today. Okay. Because if I start HR normalization today, anyway, I won't finish. So that is fine. HR in total would be about an hour. So we can do it tomorrow. Absolutely not a problem. And we can actually start with the enrichment analysis theory tomorrow. Okay, that will ease us a little bit. Okay. With that said, I am done with DC2 for the day. Do people have any questions? No questions? Anybody?
Sir, I have a question from yesterday's lecture.
Okay, please go ahead.
So now we were calculating the B0 and B1 values from the when we
Correct.
are trying to plot the linear model, right? So
Correct.
in that while calculating B1, we were using the ln scale, that is logarithm base E. But
No,
while
we were always using logarithm, the natural, not the natural log scale. We were always using logarithm base 2 or base 10. I'm not sure what I did in theory. I think it was base 10. Let me check it out once. Here. Yes. See, we are using the normal log scale, not the natural log. It
Okay,
is
so
not the natural log.
I thought so because the value 4.61 was the log 100 base E value, so I thought so.
Oh, is that so? Then
Yeah,
that might be a calculation mistake on my end. Then that is a calculation mistake on my end. Okay. It is not the natural log.
yeah, okay.
It is always so basically it is the log base 2 value. Okay. This is not the natural log, it is the log base 2 value. This is a calculation mistake.
Yeah, okay. But and one more question was that so we were looking at can you go to the previous slide in that same?
Yes.
No, so
This
the
one?
yeah, the pipeline till when we were trying to fit the model, before that we will estimate the dispersion value, right? But we were not including the dispersion for fitting the linear
No,
model. Why?
so when you calculated the dispersion value, right? If you remember, when we did DC2 today, we saw something, right? When it performed the analysis, final mean dispersion, gene-wise dispersion estimates. So when it calculates the final dispersion estimates, it adds them up to the counts. That is why we also extracted the normalized count separately, right? You remember?
Yeah, so the
Extracted the
normalized
normalized
counts
counts
is
separately.
based on the median of ratios. Is that the normalized count?
Yes, the normalized counts are based on the median of ratios. On top of that, you also add in the dispersion estimate. And then using those counts, you perform this linear model. This one, right? That is how DC2 does it.
Okay.
Yeah.
Yeah, thank you.
Yeah. Okay. Anybody else? So you are the one who texted me, right?
Yeah. Actually, yeah.
You have any other questions apart from these?
Actually, I also sent a picture showing a graph of the B0 and B1 value. So if we were looking at the fold change with a base value of 2, then the values would be different. And then, so I mean, I don't know how to put it now, but I have written that in
But
the paper.
your confusion is clear, right? I will
Right.
go through your text anyway.
Yeah.
But yeah, so a very good point you pointed out, Pavitra. This everybody, guys, in DC2, as you would have seen already, the values that we get here, let me quickly show you. The values that we get here are log base 2 values, okay? These are log base 2. So in this example, this is the log base 2 calculation. This is a calculation mistake on my end. Okay, that should not happen. Okay, it will be 2 basically. Yeah.
Yeah, thank you.
That makes sense. Sorry, not 2. It will be something else. I forgot. It will be something. If it was log base 10, it would have been 2. Yeah, it will be something else. This is a calculation mistake. Okay. So thank you, Pavitra, for pointing this out. Thanks a lot.
Yeah, thank you.
Okay. Any more questions from anybody else? If not, I guess we can wrap up then for the day. On tomorrow's agenda, we will finish Agile quickly in the first 1 hour, and we will start with the